Good morning, students. Genetics is not so easy to understand through online classes. So, if you find any difficulty in learning genetics, you can call me or I can repeat the topic once again when we meet after lockdown. Let us learn about interaction of genes. Supplementary genes and dominant genes are examples of interaction of genes. Introduction. According to Mendelism, a character is controlled by a pair of genes. The monohybrid ratio is 3:1. Dihybrid ratio is 9:3:3:1. And the trihybrid ratio is 27:9:9:9:3:3:1. The post-Mendelian studies have shown that a character need not to be controlled by a pair of genes always. However, a character is controlled by more than a pair of genes, or a single gene may control several unrelated characters. Some genes may modify the expression of other genes, some may supplement one another, and some may complement one another. Some genes may inhibit a character, some may bring about lethality, and therefore Mendelism is no more universal. Thus, it may be an exception than a rule. As such, the Mendelian ratios stand modified. Therefore, a character is a net result of interaction of many genes, and thus is called factor hypothesis or interaction of genes. Some important forms of genetic interactions are as follows. Number 1, supplementary genes. Number 2, dominant epistasis. Number 3, recessive epistasis. Number 4, polygenic inheritance. Number 5, lethal genes. Next, coming to supplementary genes. Supplementary genes are two independent genes which supplement one another to produce a novel phenotype or new phenotype. I repeat the same. Supplementary genes are two independent genes which supplement one another to produce a novel phenotype or new phenotype. When they are together, they interact to produce a novel or new character or phenotype. When they are absent also, a novel phenotype is produced. Thus, supplementary genes may be defined as two independent pairs of dominant genes which interact in such a way that each dominant gene produces its effect whether the other is present or not. But when the second dominant gene is added to the first, a new character is expressed. It is a non-allelic gene interaction. I repeat the same. Supplementary genes may be defined as two independent pairs of dominant genes which interact in such a way that each dominant gene produces its effect whether the other is present or not. But when the second dominant gene is added to the first, a new character is expressed. It is a non-allelic gene interaction. Example, inheritance of Coombs in fowls. The inheritance of Coombs in fowls was first studied by Bateson and Punnett. There are two different Coomb patterns in fowls. In Plymouth variety, there is a rose comb pattern, and in Leghorn variety, there is a pea comb pattern. Rose comb is due to the presence of two dominant genes, capital R, and P-Cumb is due to the presence of two dominant genes, P and P. But when rose-combed fowl is crossed with a P-combed fowl, novel or new phenotype walnut makes its appearance. When two walnuts are crossed in the F2 generation, walnut, P, rose and single appears in 9:3:3:1 ratio, and that is due to the interaction of supplementary genes. Rose-comb is due to the presence of a dominant gene, capital R, and P-comb is due to the presence of another dominant gene, capital P. The recessive alleles of the above genes in the homozygous condition produce single comb, that is small r, small p produces single comb. But when the two dominant genes, capital R and capital P, are brought together, they interact and produce a new comb called walnut. Walnut is due to the interaction of two dominant genes, capital R and capital P, and rose is due to the presence of gene capital R, and P is due to the presence of a dominant gene capital P, and single comb is due to the presence of recessive genes small r and small p. According to Bateson and Punnett, the combless condition is due to the presence of basic genes capital C or small c. These basic genes are modified by supplementary genes like capital R and capital P genes. When capital R gene is present, the comb becomes rose. When the dominant gene capital P is present, the comb becomes P. In fact, they modify the basic genes, that is capital C or small c. When dominant genes capital R, capital P genes are brought together, they supplement to produce a novel phenotype, the walnut comb. When they are present in recessive condition, they produce another novel phenotype called single comb. Next, coming to epistasis. Epistasis is the prevention of expression of one gene by another non-allelic gene. Epistasis means stopping or inhibiting. The inhibiting gene is called epistatic gene, and the inhibited gene is called hypostatic gene. Epistatic genes are sometimes called inhibiting genes. In epistasis, gene located on one locus interacts with another gene located in another locus. So it is a non-allelic gene interaction. Next, difference between dominance and epistasis. Dominance involves intra-allelic gene interaction, and epistasis involves inter-allelic gene interaction. In dominance, one allele hides the effect of other allele at the same gene pair. In epistasis, one gene hides the effect of other gene at different loci. Epistasis is of two types, namely dominant epistasis and recessive epistasis. Next, dominant epistasis. The prevention of expression of a gene by a dominant non-allelic gene is called dominant epistasis. inheritance of color pattern in poultry is a case of dominant epistasis. In white leghorn, there are white birds and colored birds. The colored birds are due to a dominant gene, capital C, which produces color pigments. White birds are due to a recessive gene, small c. Recessive gene cannot produce color pigments. Further, the dominant gene, capital C, is inhibited by another dominant gene, capital I, located in another locus. When dominant gene, capital I, is present along with dominant gene, capital C, the bird cannot produce color pigment and hence the bird is white. On the other hand, the recessive gene, small I, cannot inhibit the activity of dominant gene, capital C. When a white leghorn with a genotype capital C, capital C, capital I is crossed with a white Plymouth Rock with a genotype small c, small I. In the F1 generation, all fowls are white with a genotype capital C, capital I, small I. In leghorn fowl, the white color of feather is formed because of genes capital C, capital I. In Plymouth Rock fowl, the white color of feather is formed by genes small c, small I. When the F1 white fowls are crossed in the F2 generation, white and color birds appear in a peculiar 13:3 ratio. That is, 13 white birds appear and 3 colored birds appear. A checkerboard of the 16 phenotypes and genotypes of the F2 birds indicates that only 3 are colored out of 16 genotypes, that is small I, capital C, small I, capital C, and small I, capital C, small c produce colored birds. The white leghorns obviously contain a gene, dominant gene I, which in the dominant state inhibits or suppresses the expression of the dominant color gene, capital C, resulting in white plumage. Dominant epistasis is explained on the basis of interaction between a color gene, that is capital C, and an inhibitor gene, that is capital I. The plumage color is white when, number A, both color gene and inhibitor genes are present. Number 2, both color gene and inhibitor genes are absent. Number C, the color gene is present. On the other hand, the plumage color is colored when color gene is present but inhibitor gene is absent. Thus, capital C is a hypostatic gene whose expression is inhibited by a dominant gene, capital I, which is an epistatic gene. Both genes are non-allelic genes. Their interactions modify the typical Mendelian ratio into 13:3 because of dominant epistasis. That's all.